Good day, this is Professor Will, CBMD PhD. Today is Monday, July 12, 2021. It is 4:12 and 24 seconds a.m. Eastern Standard Time. Today's research, Salmonella. So let's hop to it here. New insights into Salmonella survival strategies. Date July 7, 2021. Source, European Molecular Biology Laboratory. The scientists have analyzed how the intercellular pathogen, Salmonella enterica secretes proteins to survive and thrive in infected cells, using a novel approach, which for the first time allows scientists to study which host cell proteins are targeted by the pathogen. The scientists revealed new insights into how Salmonella hijacks the cell's cholesterol supplies and how it rewires cellular transport processes to promote its own survival. Our cells fight microbial invaders by engulfing them into membrane sacs, hostile environments in which pathogens are rapidly destroyed. However, the pathogen Salmonella enterica, which grows and reproduces inside our cells, has evolved ways to detoxify such hostile compartments, turning them into a comfortable home where Salmonella can survive and thrive. Researchers had uncovered new details of Salmonella survival strategies. The researchers analyzed protein interactions in Salmonella-infected cells to identify the diverse biology processes of the host cell that the bacterium uses. The Salmonella targets and modifies cellular protein machineries and pathways in which multiple proteins work together with the help of the so-called effector proteins, which it injects into host cells. Altogether, Salmonella is known to release more than 30 effector proteins into infected cells to hijack nutrients and protect itself. However, the functions of many of these proteins and which host cell proteins they interact with are largely unknown. To find these enigmatic protein interactions, the EMBL scientists genetically engineered 32 Salmonella strains by adding identification tags to individual Salmonella proteins, earmarking one protein in each bacterial strain. The identification tags acts like a handle the scientists can grab in their experiments. This approach of modifying the effector proteins directly in their host is a breakthrough. It enables researchers to capture the bacterial proteins after they have been secreted into infected cells and to pull them out along with any host cell proteins that are bound to them. These interacting proteins are then identified using a technique called mass spectrometry. The new approach has many benefits over previous experimental strategies. In particular, it characterizes a whole set of host pathogen protein-protein interactions in cells infected with a live pathogen, closely resembling what occurs in a host organism upon a Salmonella infection. And using their novel approach, the EMBL scientists identified 421 previously unknown interactions between Salmonella proteins and host cell proteins, along with 25 interactions that had been described before. Researchers found that multiple Salmonella effectors physically interact with several proteins that the host cell uses to transport cholesterol, and in this way, cholesterol trafficking can be hijacked for Salmonella's own purposes. Cholesterol is an essential component of the biological membranes that surround our cells and the structures within them. The Salmonella uses cholesterol to modify the composition of the membrane sacs that surround it, potentially making the membrane more rigid and reinforcing the barrier that separates Salmonella from cellular detection and defense systems, which are present in the host cell's cytoplasm. The scientists also found new clues to how two other survival strategies work. One of these strategies is to remodel the network of protein fibers that are used to transport material within the cell. Another strategy involves interfering with the function of the host cell protein that mediates contacts between membranes to facilitate the exchange of lipids and small molecules. Both strategies may help Salmonella to strengthen its protective membrane shield and avoid detection by the host cell's defense systems. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.